To miejmy nadzieję, że jesteśmy gotowi. Jeszcze troszkę poczekamy. Ja może spróbuję uwspólnić ekran. A teraz screen widzicie, tak? Z napisem Lecture 2? Part 1?
Tak.
OK. Czyli wszystko gra.
Teraz.
Okay, I think we can start. So today we are going to talk about Just, I don't know if it works right now. Okay, about the replicon model, okay? So this is the concept we discussed during last lecture from early 60s, last century, and this is just something which we should remember about the concept of replicon as a basic replication unit. And we stated that for the replication initiation, initiation gene is required, and which encodes the protein, which is called initiator or replication initiator, DNA replication initiation protein. This is the term, and DNA replication initiation protein or initiator has to interact replicator, which is origin of replication. We also stated that it's just a little more complex, that we have more than one replicator and more than one initiator, more than one gene for initiator, and many initiation proteins, DNA replication initiation proteins have to interact with a particular site on the DNA just to initiate DNA replication. And the first question we would like to ask is how to identify replication origin. So how to identify replicator. It's supposed to be simple, but it turns out that it took very many years just to define the site for the replication initiation, and very many different techniques were applied to do this. So we'll go through it. The first, it's clonal analysis, then 2D gels, then 3 is features, and the 4 is bioinformatics, which could be applied to identify the DNA replication initiation sites, origins of replication, DNA replication initiation. So first is clonal analysis. Anyone has a concept what the clonal analysis is? It's a general concept in biology, clonal analysis. Yes? What do you think? How we can define clonal analysis? Well, so what is clone? There is a movie, Clone Attack or something. Am I right? Attack of Clones? I don't know, it's Star Wars or something. So clones are supposed to be duplications. Does it mean that we have duplications? We may have duplications with some variants, and then we have to analyze those variants. So in general, clonal analysis to identify the DNA replication, to identify the site for the DNA replication would be like this. Let's say that this is just the double-stranded DNA or chromosomal DNA, linear or circular, doesn't matter. And then you cut this into enzyme to pieces. Okay, so you cut, and cut. And that would create the populations of fragments, maybe different lengths, but similar if the conditions are properly applied. And then you use this DNA, you introduce this DNA to plasmids, so that would be like a plasmid template. We've introduced this one, and the other plasmid, we're introducing the other one, and so. So we will have population of plasmids, and each one would contain different part of a chromosome, okay? And then what you do, by using these plasmids, you transform bacteria. So this plasmid to this one, this plasmid to the other one, and then the other plasmid to the other one, and the other plasmids to the other one. Okay? And these are clones. Okay? It's clone 1, 2, 3, 4, 5, and 10. And then you are taking this mixture and you just grow them, because at the beginning these are single cells, and you have a plate, and if there is an antibiotic resistance in the plasmid, and the plasmid DNA replication depends on this DNA fragment which was cloned, so the only possibility for the bacteria under antibiotic stress applied would be to have this plasmid functional. Okay, this one, this plasmid functional. And I should draw this antibiotic resistance in here, an application of antibiotic over the bacteria, yes? So then you have a plate and you will have a colony. And you can expect that the colony would be formed only when the plasmid is active replicationary. So that means, for example, that it contains this part, which is a replication origin. Okay, which provides the ability for the plasmid to replicate. Does it make sense? So this is clonal analysis. And then after verifying this, having this thousands or maybe hundreds of thousands of clones, then you can see I have 10 clones, and I then by sequencing can analyze that the clone contains, each of the clone contains this part of a chromosome. So most likely this part of a chromosome is a replication origin. Okay, so this is clonal analysis. Okay, so then let's move to the other method which you can use for the analysis of replication, DNA replication initiation sites. This is called 2D gel analysis. So what the 2D stands for, two-dimensional. So it's going to be one dimension and the second dimension to resolve a gel after applying samples. And the concept is based on that. But if you have DNA replication starting at that point, at the replication origin, okay, over there, then usually after a while you have something which is called replication I. Okay, so this is non-replicated DNA, replicated DNA. So these parts are going to be this one and this one are double-stranded. And here you have also early at the beginning single-stranded, but then after a while it's going to be also double-stranded. But there is replication I. This is newly synthesized DNA. You can also look at this model. If the model is DNA replication, it's going to be bidirectional or unidirectional. You have this kind of structures. So it's going to be replication I, this, and replications origins, and replication origin in here, This is just the original picture, but I will prefer this one because the origins are kind of match. And if this is bidirectional replication, the progression of replication forks are going to be both direction. So that would create this kind of a replication I, a bubble structure, and the unidirectional replication would provide the extension of this bubble only to one end. Okay, it's unidirectional. So if you would think about this concept, so thinking that you can just use restriction enzymes and let's say cut the DNA over there, and let's say over there, So in bidirectional replication, first of all, you will have like a landmark. Let's say that this is EcoR1 enzyme, and here you have Hind 3. Okay, so this is Hind 3 and EcoR1. So you have landscape and just the landmarks of the positions of the cuts, but the substrate after this cut is going to be this bubble-like structure over there. But if you would use this in the model with unidirectional replication, you will have a cut over there, and over there. So you would get actually something which looks like this. So Y structure, and this is just like a bubble-like structure. And this is just the concept of 2D gel analysis, just to identify after restriction analysis if we have bubble-like structures, extended bubble-like structures, bigger bubbles, smaller bubbles, or DNA molecules in the shape of a letter Y. So this is the concept. So you will have simple Ys. You may have also double Ys, bubbles, and asymmetric bubbles. These are the possibilities after bidirectional or unidirectional replication, progression of replication fork, and the DNA analysis of this. This graph is a little bit misleading, because when you look at the Y over there and the Y over there, it's not necessarily very true. I mean, if we would have, I don't know where to draw this, if we would have DNA length of this, and it's going to be Y, so then when the progression of replication fork is up to almost the end, okay, then this DNA fragment would be almost doubled. Yes, because that would be this length and this length. So if you would strand it, I mean, just pull the ends, and I will draw this way, as in here, it should be this long. Is it correct? Yes, so this is too short. I would just extend this arms of the Y letter. But nevertheless, we can just look at the general concepts. So the 2D gel analysis, so the first dimension separation is by mass. So if you would, let's say that we have 1 kb structures, so this is, let's say that this is 1 kb, so this one at the end would be almost 2 kbs. Yes, because it's almost doubled. Also this, let's say average size 1 kb, so this would be almost 2 kbs. Okay, this at the end. So the first dimension separation is by mass. So on the top you will finish with 2 kbs, at the bottom 1 kb, and you will have a separation of Ys and just a picture of different sizes of but no distinguish, I mean, just 2 kb of Y would be the same on the gel as the bubble-like structure. And then you can do the second dimension. So it's exaggerate contribution of a 3D shape, and then depending on the shape, so we will have simple Ys, then it creates, so the big one would be there, and then you have this kind of a slope. Double Ys, this kind, and then bubbles. So the big bubbles in here, and small bubbles in here. Then asymmetric bubbles, well, it's asymmetric bubbles, so what is this thing? This thing actually is Y, but up to this position. Oh, I'm sorry. Oh, I screwed. I did something wrong. Okay, I'm sorry. Can you see it?
Yes, we can.
Yes, we see it.
Okay. So I just touch something. Now I just want to do the full screen, but where is the full screen over there? Okay. Yeah. Okay, so this is just, okay, oh, come on, what's going on in here? Okay, so we're there, yes? So this part is Y's, so exactly as here, and this part comes from bubbles up to there. So it's like growing bubble, growing bubble. If I would collect the samples from this position, it's going to be small bubble, okay, from bigger bubble, and the big bubble, but not the biggest one, because then, because of the cut, an extension of replication fork to this direction, then we have Y's, and that's it. So actually, just looking at these pictures, you can analyze what's going on and when the replication begins. And you can also analyze the direction of progression of replication forks. And this is just the key, just to find the right restriction enzymes around the right size. And of course, what you do, you also use the radio labeling just to get the picture labeled with usually phosphor. And these are original gels. It looks like, I would say, not very easy for interpretation, but actually based on the understanding how what is there, you can find you can understand the picture and conceive the site and direct site position at the chromosome and direction of replication initiation. Okay, so that was so we have clonal analysis and 2D gels, and now features. So what are the features which can help us to identify the beginning site of replication initiation? And the first feature I would like to mention is skew of mononucleotide frequencies, or other words, relative abundance of nucleotides, or DNA strand asymmetry, or DNA walk. You can find in literature different names for the same thing, but what is the skew of mononucleotide frequencies? We can also say skew of distribution. So it's similar. So what would be the skew of distribution of mononucleotides? What do you think? Unequal distribution, or some unfrequencies of a distribution of mononucleotides. So what kind of DNA asymmetry we have in the chromosome? Well, so we can expect that if we have DNA, chromosomal DNA, okay, and if we have, let's say, the fragment of DNA from here to here, and we will read the sequence, it's going to be A, so TT, and then, for example, TT, AA, and then G, GCC, and then CCGG. Then, when you look at the number of A's on the top, T's on the top, G's on the top, and C's on the top, comparing to the bottom strand, it's equal. So there is no any DNA strand asymmetry. But it was found that this is not the case in the chromosomes. So if we would take the DNA fragment and then we would do this analysis in particular parts. So in one part we will have A, T, 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 but only one T on the top and then only one A on the bottom. So that gives asymmetry between the pairs distributions, or rather particular nucleotides distributions between the top and the bottom strand. And this distribution tells you where the termination site of a DNA replication and origin of a replication is. So because the termination and the DNA replication sites are these extremes with unequal distribution between A's and T's and G's and C's. And here you can see this deviation form between the A's and T's and C's and G's. And so the relative nucleotide skew that are normalized differences in the content of complementary nucleotides change the sign crossing replication origin. So this is just the replication origin and termination. And so if we would go and analyze the sequence of chromosomes, circular chromosomes, you will find this asymmetry. And based on this, you can expect that the position of replication origin. Okay, so this is one of the features. So what other features you can identify to identify the position of replication origin in bacteria? Well, these are AT-rich regions and DNA boxes. So AT-rich regions are regions which have higher than average contents of A's and T's. So it's like less stable part of a chromosome. And usually those regions are also have directed repeats. And the other feature is DNA box sequences. And DNA box sequences are binding sites for DNA replication initiator. The DNA replication initiation protein is usually not only replication initiator, but also transcription regulator. So you can find the DNA boxes all over the chromosomal DNA. However, the concentration of DNA boxes, so binding sites for the DNA replication initiation proteins, are close to the replication origins. This is just original electromobility shift assay or gel mobility shift assay showing the complexes of DNA A protein with DNA. So you can identify these binding sites by just using in vitro assay with DNA fragments, and you can just look at the retardation of those fragments in electric field due to the complex assembled, different complex assembled by DNA proteins, and it was just found in 1996. The other technique just to identify the interaction of a protein, whatever protein it is DNase-1 footprinting assay. So it's an enzymatic reaction in which we can just footprint, so analyze and identify the site of the interaction, and therefore the sequence which is recognized by the protein to interact DNA. And this is just how you do this, and this is just the picture of a gel, and you can see bands and some kind of lighter regions of a gel which is protected by the protein interaction, DNA interaction, okay, in these sites, and this is just protected from the degradation by enzyme, which is called DNase-1. And you can see on the right side of this slide how it is, how this experiment is performed. So usually we label one end of analyzed DNA fragment, and this fragment has to be, I don't know, in hundreds of nucleotides long. And then you add the protein, so in there, okay, and this is just the protein complex, protein DNA complex is formed. Then you add the DNase-1 and you perform incubation for the digestion, and the DNase-1 is supposed to cut the DNA, but it cannot cut in the site when the protein interacts the DNA. So actually you collect the hundreds or thousands of DNA which is labeled at the end and cut it usually in one side. So it's labeled, let's say cut it in here, then something labeled it, cut it shorter version, yes, and then you have something like this, but that would be no fragments protected by the protein A interacting in here, because nuclease cannot cut there, okay? So you will have these fragments and longer ones because of cuts in there, but not this length, and this is this part. Okay. So this is just DNase-1 footprinting and EMSA or gel mobility shift assay to analyze the site of the interaction of a protein. So what we can get from the site of interaction? What kind of information? Of course, if you had this kind of experiment, then you can just run the ladder identifying the position of cuts, and then you say you can get information about the sequence which was protected, and based on this analysis you can find the composition of binding site of particular protein, in this case the replication initiator, so DNA A, and this composition of DNA A, so-called boxes. And this composition is described as a DNA box consensus sequence. Okay, so you can find consensus sequence for binding of any protein. And what is the consensus sequence? Consensus sequence, in Polish, we should say, please don't say sekwencja konsensusowa, because it doesn't make any sense in Polish. Mówimy, so we say sekwencja najwyższej zgodności. This provides the probability that the protein would interact the piece of DNA with T at the beginning. Let's say that this is 93.7% probability. Then the other position is also T. Yes. Then we have A or T. And then T. N stands for no matter what. I mean, no, maybe not no matter what, but any nucleotide. Then C, A, C, A, with some probabilities as well. Okay, so that gives this consensus sequence. Sekwencja najwyższej zgodności. Okay, so if knowing the consensus sequence of a replication initiator, then we can identify DNA box sequences all over the chromosome, and then we will have the information about the distribution of those DNA boxes. And the highest distribution, highest concentrations of DNA boxes in particular site would most likely provide an information of DNA replication initiation site. And this is just an example of this kind of an analysis. So we have Streptococcus pneumoniae chromosome, and you see that this is just unequal distributions of A's and T's, pointing this part as a most likely replication initiator. Then the DNA boxes positions, so this is just the numbers of DNA boxes all over of a chromosome, and you see then in here we have a concentration of DNA boxes, and the red part is the position of a gene for DNA A protein. It's also close to the replication origin. So if these three features matches, okay, then you can most likely say that, okay, this is just the position of replication origin. But the real life is different, and you can see these are pictures from different bacteria. So as in here is a perfect match, but sometimes not necessary. So in this position you can see that the skew and the DNA boxes are fine, but the position of gene encoding DNA A protein not necessary. Then we have some bizarre number of DNA boxes in this part, number C. Then you have some kind of twist position of DNA box concentration, cumulation, and position of gene for encoding DNA A protein, but and you see that this is just, it varies, but nevertheless, to some extent, it's helpful to identify the position of DNA replication origin. Okay, I mentioned also this A plus T richness, that this is just something very typical for the replication initiation sites. So intuitively, you can say that just to opt for the replication initiation, you have to destabilize double-stranded DNA. So it's easier to destabilize something which has adenines and thymines pairs, not GC pairs. So this is the reason that within the replication initiation origin, we have this AT rich part, and this is called also DUE, so the DUE, no matter what chromosomal, extrachromosomal, prokaryotic or eukaryotic replicons, stands for DNA unwinding element. And this DNA unwinding element is typical for replication initiation origins, has lower, higher than average AT content, and also some kind of fluctuations and repeated sequences inside, which are marked in here by this asterisk. Yes, and you see those fluctuations of thermodynamic stability. So you see that there's just up, down, up, down, up, down, up, and then minimum up, down, up, down, up, down. So not only that there is some kind of a minimum, but also fluctuations are typical. So where those fluctuations come from? Well, based on the sequence, because most likely you may have AT, TT, and then GC, GC, and then TT, TTAA. So if you would draw this internal stability, that would be like a very low, then a little bit bigger, and then again very low, and that makes those peaks. Yes? Okay, so this AT richness could be identified just by sequencing the genome, but also it can be identified by the AT richness could be identified, but not necessarily DNA unwinding element, because it's something different. The AT rich part of a chromosome could be identified by the sequencing method to identify if this particular sequence of this AT richness is unwind during replication initiation. Is not that easy, and it requires experiments. And what kind of experiments you can perform? There is the second type of footprinting analysis, which we are mentioned right now, and this is called P1 footprinting. So the previous one was DNase 1 footprinting, and P1 footprinting is a little bit different. The DNase 1 footprinting is used to analyze the interaction of proteins with DNA, and P1 footprinting is used to identify single-strandedness of the DNA. So, well, you have to add the proteins, replication initiator, to the complex and form the DNA protein or protein DNA complex. And due to the protein activities and the activity of a replication initiator, the DNA unwinding element is going to be unwind. So unwind, it means the single-stranded DNA is going to be present in here. And since P1 nuclease has an affinity for the single-stranded DNA, it would cut this single-stranded DNA. Okay, so these cuts are over there. So after this kind of a reaction, we can perform primer extension. So what is primer extension? Primer extension reaction is the reaction when you take the template, anneal the primer. Usually the primer is labeled, okay? Then you add so it would provide free prime end, and then you add polymerase to extend it. So that would be extended up to the end. So actually in our experiment, it would be extended to the place when the P1 digested, where the P1 digested DNA. So actually this length of a synthesized strand would identify the position, yes, of which is unwinded. Okay, maybe I just control. So let's say that we have this replication bubble, but not this later on, but this before the this is just a single-stranded, early at the beginning, okay? Single-stranded DNA. And this is just due to the protein interaction. So then you will have the cut of the single-stranded by P1. And then in the solution, you can anneal the primer, extend it, and identify the position of the cuts. And since those cuts would be like each molecule, you should just optimize the reaction that each molecule in your reaction would be cutted only once. So one in here, the other one in here, the other one in here, but not in here, not in here, and not in here. Then you will have something which is over there. So bands on the gel. And those bands on the gel, yes, would represent the fragments cutted by P1 nuclease, and then you can just identify the length and the position of those cuts. Okay, so this is just comparison, just not to make you difficult to remember and understand. The P1 for single-strandedness identification and DNase-1 footprinting to identify the protein interaction site at the replication origin. Okay, so I think we can do a little bit break, 10 minutes. Yes, so let's meet at 1:10.
Can I have a question?
Yes, please.
So what's the difference between replication initiation origin and the DNA element? Like, because I understand the function, but are they both just AT-rich fragments, or is there any difference between them?
Well, so replication origin is something bigger, and DNA unwinding is a part of replication origin. Let's say that if I would draw this, and I would say, so this is the replication origin, and it would be like binding site of a protein, the other, what happened? The other binding site for the protein, then the other binding site of a protein, and then something which we would say DUA, DNA unwinding element, and then binding site for the other protein, let's say FIS, and so and so. So this is just the core, the place when this original unwinding, which you can identify with using P1 digestion, would happen. But if you would use this DNA unwinding element and you would try to create, let's say, artificial origin, that would not work, because that would be not other factors, like, for example, binding sites for proteins required for full activity of replication origin, okay? So this is just like a part of replication origin, this DNA unwinding element.
Yes, but it only consists of AT-rich fragments.
Yes, AT-rich fragments, and usually, well, you can have some overlaps, and sometimes it has repeated sequences inside, and this is just repeated, are short fragments. I will probably talk later on a little bit about this, but these are like short repeats up to several nucleotides repeatedly and directed in one direction. And this gives this instability and those peaks which are talked about.
Okay, thank you.
Okay, so 10 minutes break, yes? Okay. Maybe right now. Can you hear me right now?
Yes.
Okay. I have my microphone off. I need to collect your attendance. Okay. And let's continue. Okay. So based on those data, okay, about the P1 footprinting and DNA SWAT1 footprinting and gel retardation, okay, does it work? The model of replication initiation protein interaction with origin, replication origin was proposed. You see, this is from the Arthur Kornberg laboratory. Well, it was that time that we had kind of like a bubbles, black bubbles, and some kind of wrapped around DNA sequence with 9 mers for the consensus sequence interaction site for DNA A protein, and single-stranded DNA in there created by the interaction of DNA A protein with DNA, and so-called open complex structure and initial complex. So initial complex is just the initial binding of a replication initiator, and open complex is just something which was created, this initial bubble with single-stranded DNA. Okay, however, it was found that the interaction of DNA A protein with DNA is the nucleotide dependent, and depending on the ATP or ADP you see on the electromobility shift assay, that this picture looks like a little bit different. So we can identify in here bands of very many molecules of DNA A protein interacting with very many sites on the DNA, but those bands are at least barely or not present when the protein was incubated with ADP instead of ATP. And this is just also DNase 1 footprinting, showing this ATP-ADP dependence. You see that in the presence of ATP, this footprint looks better comparing to the presence of ADP. And the next experiment is surface plasmon resonance, that on the sensor chip we can attach the double-stranded DNA, and then we can identify the interaction of the protein with this attached DNA fragment. And you see that depending of the sequence of nucleotide attached, we can identify the resonance signal. Okay, this is a resonance unit, and this shows the mass distribution of a mass in time interaction of a protein in ADP and ATP form. This is very sophisticated. It uses specific sensors. We have a gold dextran surface, and on this, so it's a glass, the blue thing, and then you have a gold layer, and on this gold layer there is dextran surface where you can attach DNA or proteins and then look at the interactions. And the interactions, it's okay, everything is just in the chamber. So this is just the sensor chip. Okay, this is just the gold layer. Then you have your ligands attached, for example, DNA, and it's a flow channel. So you can pass the buffer and you can look at the interaction of the proteins in here. And since there is a flow of a buffer, you can just see when the proteins or molecules are interacting, what is attached to the sensor, and then when the interaction is released, so you can also detect it, and this is reflected by the optical detection unit with a light source and polarized light, and you can see different angles with when the mass on the surface is changed due to the interaction. This is just the basis on the surface plasmon resonance. So someone can ask, so what is this plasmon? So the plasmons are specific electrons which actually are created due to the change of the mass on the gold surface. So when the mass is changed in the channel, then those electrons are activated, and then this changed the reflected light angle. And based on this, we can just detect the sensogram and look at the mass attached in the sensor. And based on this, it was possible to identify the boxes by the DNA, but there is a special one. So far, I told you about the consensus sequence, and this is just the other consensus sequence, which is ATP dependent. And you see that those boxes are a little smaller, and they are present within the AT-rich part of replication origin. So this is our box, which is outside DUE, but those boxes you can identify within the DNA unwinding element. So this is just the comment considering your question about the structure of this DNA unwinding element. So there are repeated sequences, but also those repeated sequences you can see over there. But also specific binding sites for proteins. Could be. But based on those experiments, the other consensus, ATP DNA box consensus, was identified, and it was found in very many bacterial chromosomes and extrachromosomal genetic elements. Well, so that was in Walter Messer laboratory in Max Planck Institute for Molecular Biology in Berlin. He proposed that DNA protein can bind DNA boxes which are outside AT-rich part of replication origin with those repeats, but also it forms some cooperative kind, low affinity cooperative DNA binding within the AT-rich part of replication origin. Okay, and this cooperative binding with high affinity cooperativeness between DNA molecules creates this origin opening, so single-stranded DNA. Okay, so the situation is more complex. The binding sites, we have two different types of binding sites by DNA protein, by replication initiator. One, just let's say there's traditional 9 nucleotides long with a consensus sequence 9 nucleotides long, and shorter one with which requires ATP for the DNA interaction with those sites. Okay, this is just these conclusions, and I think we can just move to the part 2 of this lecture, and it's going to be about the structure of DNA protein and how it looks like and what is the current knowledge about the structure of a replication initiator. Well, you can see alignment with of the DNA sequence from different bacteria species, and of course you can find some similarities and differences, but no matter what, it is accepted that the protein consists of 4 domains. Domain 1 with Leuzer zipper and different helices, helix 1, helix 2, helix 3. Then part 2, domain 2 is less conserved among species. The part 3 in here with a very unique structure having Walker AB motifs, also helicase DNA-B interaction site, and ATP binding. So this, if we have ATP binding, you can assume that this is the region of a protein which we called AAA+, because this is the activity associated with ATPase, different activities associated with ATPase. And the 4th domain, the DNA binding domain, which forms the helical bundle at the C-terminal end of a DNA protein. Okay, so this is just the sequence and the structure, but it was very many attempts to obtain the crystal structure, and finally it was achieved in 2003. And of course, when the people tried to get the crystal from E. coli, which was E. coli DNA protein, they failed due to some intrinsically disordered regions of this protein, but then you can just move to thermophilic bacteria, and it was done, Aquifax aerolicus, and you see that this is just the structure of a protein, so the AAA+ is there, so this is this part, yes, and DBD stands for DNA binding domain, is going to be this part. Okay, so this is just the thing. And what you can also notice in this, the red thing in here, what you expect is this. In the AAA plus. Well, it's ATP, okay? So this is the site for the interaction of ATP. Okay, but this is just the monomer of a DNA A protein, and the question was, so how it works, and what is the, why we have so many DNA A boxes in the replication origins. And it was very many years. I mean, maybe not that many, but still we have, you will see some misconceptions and discussions about the structure of a DNA A protein. But it was proposed by Berger Laboratory in May 2007 that the DNA A protein in ATP form forms helical filament. And this is just the filament structure on the DNA. Yes, and this is just the top view. And this is just the model of this. So it was proposed that this filament can kind of like bend the DNA, and this bending results with the opening of a structure. So not only just simple interaction with one side, but all DNA A binding site has to be occupied. And then there is a debate if the protein has to interact with this single-stranded or not to create opening reaction. And these are the nucleotides, amino acids, which interacts DNA. And this is just particular protomers of DNA A protein, so DNA binding domain, AAA plus, AAA plus interaction, DNA binding domain, AAA plus interaction, because within this filament we have some kind of a domain sharing, so one DNA A protein molecule has to interact the other DNA A molecule within the filament to form the filament and to interact the DNA. This is the other model. The model proposed compact and extended structure of DNA A protein and interaction. So this is just the DNA and interaction within the extended monomers of DNA. Yes, so you expect that this is this interaction point, but then after this compact arrangement, everything is very close to each other. And the Japanese group worked on the Motoga maritima DNA A protein, also found that specific mutated versions of protein within the AAA plus domain affected the interaction of a protein with DNA, and those mutations in their model were faced to the channel formed by the filament. It looks from in here as a channel, but you see that this is kind of like extended, as this filament forms like extended spring. Yes, and those residues listed in here resulted with non-functional protein, and non-functional in terms of the open complex formation was not present with those mutants. So this initial unwinding of double-stranded DNA was impossible. So specific amino acids in each protomer of DNA A face to the DNA, and this interaction is important for the open complex formation, and this ATPase pore of assembled DNA binds single-stranded DNA. So this is the interaction with single-stranded DNA of those protomers and this filament structure of DNA protein. And again, the residues facing to the inner channel. This is more simple model, and as I mentioned, still there is debate if this AAA+ interaction with single-stranded DNA is extension of the filament. Okay, so this is the filament form, and could it be extended, or this interaction is the separate interaction of a protein not necessarily related with this filament formation on the DNA. So we have filament and then extended and then compact form, and the compact form results with the opening of a replication origin. Well, so we kind of moved within the how many years? That's okay. So Kornberg experiments. So this model was proposed in, I think, 80s or maybe 90s. And now we have next century, so 50 years or so, to get the picture of a DNA filament on the replication origin, but still we don't know everything. So this is just the current model, and just to prove that we don't know, I would like to show you the Nature paper from 2016. So we thought that actually everything is discovered based on the sequence of replication origins in bacteria, and then this paper showed up that the new specific sequence was identified, which is called DNA trios. So this is just the trios which were identified between DNA box sequence and the AT-rich part of replication origin. So this manuscript demonstrated that those trios are absolutely necessary for the activity of replication origin. And these trios are also required for the interaction of DNA protein protomers 1, 2, 3, and 4 with single-stranded DNA. The authors could not detect the interaction with single-stranded DNA when they just mutated or deleted those trios from the replication origin. So still you see that this is just something which still could be discovered, and something which we would not expect, that just repeated sequences would not be identified up to 21st century within the bacterial replication origins. Okay, I think, okay, maybe this one just not. I would just put, okay, this is just the proteins, which, so I mentioned that these AT-rich regions are important for interaction of a DNA protein. However, it's not only DNA protein which interacts, and then we will talk later on, but also DNA C, DNA G primase, polymerase subunits, holoenzymes, single-stranded binding proteins, SEC-A, EGA, Pi from plasmids, FIS from plasmids and E. coli, IHF protein, HOP, and other proteins are interacting these 80 rich regions of replication origins, and majority of those interactions have a regulatory importance. And I think during next lecture we will talk a little bit about this regulatory importance. Okay, so that would be it for today. Yes. So, do you have any questions? Just to summary. So, replication origin is not that easy to identify. We specified different ways to identify the techniques to identify the replication origin, starting from clonal analysis, 2D gel analysis, and features of replication origin, sequence data, and different experiments which can help you with this identification. Also, we talk about the identification of DNA binding sites and consensus for the consensus sequence for the interactions of replication proteins within the replication origins. We talked about the open complex structure of DNA protein with different domains of the protein, and the quaternary structure formed within the nucleoprotein complex at the replication origin, which is considered as a filament with specific interactions with both single-stranded, and I should say double-stranded and single-stranded DNA. And then the importance of interaction with single-stranded DNA is ATP dependent, and also depends on this specific sequence, consensus sequence, short ATP DNA boxes within the AT part of replication origins. We also mentioned some kind of concepts as a DNA unwinding element, DNA boxes, AT rich part of replication origins, molecular skew, so unequal distributions of nucleotides on the top and the bottom strand, and at the end trios, which are also necessary for the DNA replication activity and initiation of DNA replication. Thank you very much, and see you next time. Yes, are we meeting next week? Yes, we should. Okay, the first is on Wednesday. Thank you, bye-bye.
Thank you, bye.
Thank you.
Thank you. Bye.
Bye.
Thank you. Bye.
Bye.